Uh, in this module, we will discuss that how antibodies can interact with the small molecule and how antibodies can be made to a smaller size to a smaller molecule. Because antibodies, they are after all proteins and they have long structure. Now the immune system, it recognizes the foreign proteins. You see, this is the basicity of immune system, that it recognizes the foreign proteins and peptides if they are present. Now, if they have a molecular weight which is greater than 2000 Dalton, then the immune system will be able to recognize these proteins and peptides. Now, this magnitude of response will increase the greater molecular weight. Now, if the molecular weight is greater, then the response of the immune system will be greater. If it is less than 2000 Daltons, then it will be lesser, the immune response. Proteins and peptides, generally they have a molecular weight greater than 2000 Daltons. Almost all proteins and peptides, they have a molecular weight greater than 2000 Dalton. And all. Now, if an antibody is to be made to a molecular weight smaller than 2000 Dalton, then it has to be conjugated. That if you want an antibody which has a molecular weight less than 2000, then you have to conjugate it to a certain other chemicals. Now, these small molecules, they are known as heptines and maybe peptides, organic molecules, or other smaller chemicals. So you can interact or you can combine those peptides to these small molecules, the antibodies, to make them less than 2000 Dalton. They are usually conjugated a protein such as albumin, such as keyhole limpet, hemocyanin, or the thyroglobulin, and then used to immunize animals for antibody protection. So what you do, you conjugate the protein to these three conjugants: albumin, keyhole limpet, hemocyanin, and the thyroglobulin. And then you immunize with these compounds to the animals to produce the antibodies. Just as shown in this diagram, that heptine molecule, that is heptine, is not recognized by the immune system alone, then the heptine, it is recognized by the immune system when it is combined with the antibody, so that its molecular weight increase. Now, if a polyclonal antibody that is being made, it is advised to change the carrier protein at least once in the immunization procedure. That if you want to make a polyclonal antibody, then the carrier protein during the immunization you have to change in the procedure once in the cycle. Now, this favors more antibody being made to the heptine and less to each of the carrier proteins. That is, you have to change the carrier protein once or while. Now, if a monoclonal antibody that is being made, then the carrier protein can be the same throughout the immunization. If you want to, however, make a monoclonal antibody, then the carrier protein can be of the same throughout the immunization procedure rather than changing them. In case of screening hybridomas for monoclonal antibody production, you see we are producing the hybridomas as well, which we have discussed earlier. Now, if we have to screen the hybridoma for monoclonal antibody production, it is necessary to screen against the heptene and the carrier protein. So you have to screen them against the heptene and the carrier protein, which you have made separately. Why? We will discuss here that any antibody which responds to the both. Now see that if we do the cell test and it is responding to the both tests, then what will happen? That any antibody that is responding to the both, that should be discarded, as there will be recognition the junction between the heptene and carrier. You see, those antibodies, if they are the same, that they are showing the same results in this case, that should be rejected because they will be recognizing the junction between the heptene and the carrier, which we don't want. That only the junction between the heptene and carrier, that will be recognized, and it will not recognize due to the native heptene. It means that in our test, they should be respond only to one rather than the both. Because if they give the response to the both, that will, as a matter of fact, recognize the junction between heptene and its carrier protein rather than the antibody which we are producing.